Are we good to go with the live stream? Are we good to go? Okay. All right, guys, so hopefully you guys are all here for Bio 2 final exam review. Yes. If you were not able to find the practice test, it is on this website under the biology tab. So we're going to go through that today and just ask questions as we go, and then at the end we should have some additional time to answer questions that you guys have. This session is being recorded and live streamed, so just be cautious of like background noise just for the video. Are there any questions about anything before we begin? Was everybody able to find the practice test okay? Okay, cool. All right, so let's get started. So for number 1, which of the following is false regarding the endocrine and exocrine glands? What did we think for that question? Okay, so we have one saying D, both A and C are false. Does anybody disagree with that? No? Not that one? What do we think? Okay, so let's go through them. So the exocrine glands secrete hormones for communication within the body. Is that true or false? True, false? That's false. Why is that false? Yeah, so that's what the endocrine glands do. And what about B? Endocrine glands secrete hormones into the external environment. Is that true or false? True? Does everybody agree with that? False? Why is that false? Awesome. Yeah, that's what the exocrine glands do. And endocrine glands have ducts, while exocrine glands do not. Is that true or false? True? False. Why is that false?
Because the endocrine is the one that are ductless, and the exocrine is the one that has the ducts.
Perfect. So that's false because it's the opposite way around. So that means our answer is going to be E. All of the above are false. Is everybody good with that question? All right. So which of these is an endocrine function? The liver secretes bile into the lumen of the bile duct. The kidney secretes urine into the ureter. The liver secretes thrombopoietin into the blood. The pancreas secretes bicarbonate into the lumen of the pancreatic duct. Or none of the above are endocrine functions. C, the liver secretes thrombopoietin into the blood. How do we know that's the correct answer? Awesome. So this is a hormone. And if we did not know that was a hormone, what is the other clue in that question that we would know that was an endocrine? The liver. So the liver is an endocrine gland. But the main point of this one is that this is the only one going into the blood. So the endocrine system always secretes hormones into the blood. So even without knowing that the liver has an endocrine function and without knowing that this is a hormone, we would know from the fact that this is going into the blood that that was the correct answer. Okay, is everybody good with that? Cool. All right, neurohormones are specialized types of hormones that do what? So do neurohormones cross synapses to continue electrical signals? No, perfect. So we know that's not the answer there. And are neurohormones released from specialized types of neurosecretory neurons in the blood? No? What do we think? That's true, perfect. And are neurohormones local regulators? Yes? So let's think about our endocrine system and hormones in general. So neurohormones are specialized types of hormones, and all hormones going to be local or distant regulators? They're distant, right? Awesome. So that means that B is going to be our answer there because these are distant regulators. Okay. Everybody good with that one? Okay, cool. Okay, what is one possible consequence of a low iodine diet? What do we think? So if we have a low iodine diet, what does iodine do? What do we need iodine for? Awesome. So we need iodine to make thyroid hormones. So if you have a low iodine diet, that means you're not going to be able to make your thyroid hormones. So are we going to see hyperthyroidism due to the thyroid releasing an excess of hormones? No, awesome. And are we going to see hyperthyroidism due to the thyroid releasing too few hormones? No, because hyper is the wrong one. That's not the one we're looking for. And so are we going to see hypothyroidism due to thyroid releasing excess hormones? No, because we're not going to see an excess of hormones. So that means we're going to see D, hypothyroidism due to the thyroid releasing too few hormones. Mm-hmm. Exactly. So hyperthyroidism would be the opposite. Exactly. All right. Any more questions so far? No? Okay, cool. And these are all from chapter 45 so far. This whole page is from chapter 45. Okay. So which of these sex hormones is primarily produced by the gonads and is present in both males and females? Androgens, estrogens, progesterones, androgens and progesterones, or all of those? Any ideas? Mm-hmm. Perfect. That's exactly right. So all of these are going to be produced by both males and females, but they're going to be produced in different proportions. Any questions about that? Peregrine and autocrine chemicals are part of the endocrine system. Is that true, false? Are peregrine part of the endocrine, or are autocrine part of the endocrine? True? What does everybody think? Is that true? No? That's actually going to be false because the endocrine system are distant regulators and peregrine and autocrine signaling are local regulators. So these are both a form of local regulators. And they're also not, they're not dealing with the blood like the hormones of the endocrine system. Awesome. Any questions about that one?
So what are they categorized at, like besides local regulators, if they're not part of what are they?
They're just classified as local regulators. So number 7, the adrenal cortex and the adrenal medulla differ in that it's kind of a long one. So does anybody have an idea for number 7? So let's look at them one by one. So the adrenal cortex reacts to endocrine signals and results in long-term stress response, while the adrenal medulla reacts to nervous input and results in short-term stress response. Is that true? I see you nodding. So we've got one that thinks that's true. Awesome. What about B? The adrenal medulla reacts to endocrine signals and results in long-term stress response, while the adrenal cortex reacts to nervous input and results in short-term stress response. Does anybody think that one's true? No. Awesome. The adrenal cortex reacts to nervous inputs and results in long-term stress response, while the adrenal medulla reacts to endocrine signals and results in short-term stress response. Is that one true? No, that one's not true. And the adrenal medulla reacts to nervous input and results in long-term stress response, while the adrenal cortex reacts to endocrine signals and results in short-term stress response. Is that one true? Awesome. So does everybody agree that A is our correct answer, or does anybody think that E, none of the above, is correct? Yep. So A is going to be our correct answer there. Everybody good with that one? And the answers to this are going to be posted on the Study Union website after this session as well, so in case you lose something. Okay. All right, so let's look at number 8. The adrenal response to releasing epinephrine in response to stress is an example of signal transduction, an external trigger, an internal trigger, both signal transduction and an external trigger, or both signal transduction and an internal trigger. What do we think? Mm-hmm. D, awesome. So why do you think it's D?
Because it's a signal transduction, it's an internal trigger.
Okay, good. All right, so you are right. So it is D, and this is also in chapter 45. So what is signal transduction? Does anybody remember that? Mm-hmm. Awesome. That's exactly right. So we're converting extracellular chemical signals to get a specific intracellular response. And so we know that this is going to be an example of an external trigger, the adrenal response of releasing epinephrine in response. Can anybody think of an internal trigger that we talked about in class? So one example of an internal trigger that he talked about was estradiol binding to the cytoplasmic receptor to form a hormone receptor complex. Does that make sense? All right. Any questions on that one? Okay. All right, so now we're into chapter 46 questions. So fragmentation is a form of asexual reproduction in which new individuals split off from existing ones, and egg remains unfertilized and results in offspring. The parents separate into two approximately equal-sized individuals. The body breaks into several pieces, each of which regenerates into a new organism, or a hermaphroditic organism self-fertilizes. What do we think about that one? D? Awesome. So that's going to be fragmentation. What would this be an example of? What do we call that? Perfect. Budding. And what about B? An egg remains unfertilized and results in offspring. Awesome, parthenogenesis. And what about C? The parent separates into two approximately equal-sized individuals. What do we call that? Awesome, fission. And what about this one? What is when a hermaphroditic organism self-fertilize? What's that an example of? Yeah, so that's going to be an example of sexual reproduction. Awesome. You guys good with that one? All right, so question 10, what is a benefit of sexual reproduction over asexual reproduction? All right, does anybody have an idea for the answer for number 10? Mm-hmm. C? Awesome. Does anybody disagree with that one? No? Okay, cool. So a benefit of sexual reproduction over asexual reproduction is that due to recombination and genetic variation, sexually reproducing organisms are more easily able to adapt to changing conditions. And These are going to be advantages of asexual reproduction. So asexual reproduction requires less energy. So sexual reproduction requires more energy. And asexually reproducing organisms are able to generate more offspring in a shorter amount of time than sexually reproducing organisms. And are there any questions about that one? All right. So which of the following is a true statement regarding external and internal fertilization? So let's go through those. So organisms that fertilize externally rely on moist environments to prevent egg desiccation. Is that a true statement? Awesome. Internal fertilization allows for more sexual selection in the form of mate choice. Is that a true statement? Awesome. And the success of external fertilization often relies on synchronized release of eggs and sperm. Is that a true statement? Awesome. So all of those are going to be correct. That's also chapter 46. Awesome. All right. So what is the difference between serial and simultaneous hermaphroditism? Does anybody take a gander? A, awesome. Does everybody agree with that one? Okay, cool. So serial hermaphrodites change from one sex to another. Simultaneous hermaphrodites have both male and female sex organs at the same time. That is a true difference. Does anybody remember the other name that we can use for serial hermaphroditism? Awesome. So this is also sequential. Are you able to see okay? Am I in your way? Okay. And so if we go down to these ones where we're talking about self-fertilization, so these are wrong because most simultaneous hermaphrodites don't self-fertilize, but some of them can. So they're capable of it, but that most of the time they don't. And what type of organisms do we usually see hermaphroditism in? Awesome, sessile organisms that don't move around a lot. Awesome. Any questions about that? All right, so which of the following is correct regarding the corpus luteum? Any ideas on that one? So let's go through them. During oogenesis, it develops from the ruptured follicle. Is that correct? Awesome, that one's true. Its function is to secrete estradiol and progesterone to maintain the uterine lining. Is that correct? Awesome. And it forms in the embryonic stage of oogenesis. Is that correct? No, that's incorrect. So in the embryonic stage of oogenesis, we have our follicle, but our corpus luteum is formed in the puberty stage during ovulation. So D is going to be our correct answer for that one. That's chapter 46. Any questions about that?
For C, what stage does it form in?
It forms during puberty. So after your follicle, after the follicle ruptures, because this happens in the puberty stage, and we know that it develops from the ruptured follicle. So in the embryonic stage, you're just in the follicle, which is within the ovary. Good. You're welcome. Any other questions about that? Okay. All right. in the process of spermatogenesis, what happens directly after stem cells mitotically divide to create diploid spermatogonium? So, directly after stem cells mitotically divide to create diploid spermatogonium, what's going to happen? Will the spermatogonium undergo meiosis I to form 2 haploid sedentary spermatocytes? No. This is what primary spermatocytes do. So, does the spermatogonium divide mitotically into diploid primary spermatocytes? Maybe. That is going to be our correct answer for that one. So that's what our spermatogonium does. So we know that these are false, so how would we correct these statements? What divides in meiosis II to create haploid spermatids? Awesome. Exactly right. So these are going to be our secondary spermatocytes. And we know that that's not going to be true. So what divides mitotically to create primordial germ cells? Nothing, right? Our primordial germ cells are what start the process, right? So we're not going to see anything like that. These are going to be our first step, the first thing that mitotically divides. Awesome. Any questions about that? No? Okay, cool. So, which of the following stages of oogenesis occur in the embryonic stage? What do we think about that one? Let's go through that. All right, so the haploid secondary oocyte stops at meiosis at metaphase II and is released from the follicle. Is that in the embryonic stage or is that in puberty, after puberty? That's not going to be in our embryonic stage. This happens right after our follicle stimulating hormone comes in and stimulates the release of the primary oocyte. And mitosis in the oogonium creates diploid primary oocytes that stop at prophase I. Does that happen in our embryonic stage? Awesome. So we know that one is going to be true. And what about unequal cytokinesis at meiosis I produces a primary oocyte? Is that in our embryonic stage or is that sometime after puberty? That's going to happen after fertilization. So fertilization stimulates the completion of meiosis, and then that's where you get your first polar body that disintegrates in that step. So B is going to be the only one that occurs in the embryonic stage. Does anybody know what happens directly before this? What's the step right before this? So before this happens is when your germ cell is going to mitotically divide to create the diploid oogonium. So this is developed from our germ cell mitotically dividing, and this is the point where it's going to stop, and this is the last stage before puberty. So this is where it stops until you hit puberty. Good with that? All right, select the false statements. All right, so gonadotropin-releasing hormone is secreted by the hypothalamus and directs the anterior pituitary to secrete gonadotropins. Is that going to be true or false? True, awesome. During the follicular phase of the female reproductive cycle, luteinizing hormone stimulates the transformation of the follicle into the corpus luteum. Is that going to be true or false? False, awesome. So why is that false? So this happens in the luteal phase, and not the follicular phase. And during the proliferative phase of the uterine cycle, estradiol and progesterone are secreted by the corpus luteum to stimulate the development of the uterine lining. Is that going to be true or false? True, awesome. So that means B is going to be our answer for that one. That's chapter 46 as well. So is everybody good with that one? All right, so this is where we get into chapter 48. What happens during the falling phase of action potential? So during our falling phase, what are our phases, first of all? So we've got our resting state, depolarization, rising, falling, undershoot, right? So let's look at these. So most sodium channels close and most potassium channels open. Does that happen in the falling phase of action potential? Yes, right? And what about most sodium channels open and most potassium channels close? We know that's not right because that's the exact opposite of that. We know that's not right because we just said those two. And what about the sodium-potassium pump returns the cell to resting potential? When does that occur? This happens after the undershoot phase. So this one, we're restoring the negative charge inside. And are these sodium and potassium channels going to be gated or ungated? These, yeah, awesome, yeah. So these specific ones that we're talking about are going to be gated. But remember, there are ungated ion channels as well in the cell. Any questions about that one? Mm-hmm.
The restoring the negative charge would be for A?
Yes, so that's why this is happening in the falling phase, because in the falling phase, we're trying to get that membrane potential back, back into the negatives. Because in our rising phase, we got way up over the threshold into the positive range, and so in the falling phase, we're trying to get back into the negative, and then we hit that undershoot, where it's below the resting potential. Good. Awesome. Any other questions about that? Okay, cool. So, which of the following statements regarding neuron structure is true? All right, so do dendrites receive electrical stimuli from other neurons? Is that true? Yes, awesome. And the synaptic terminal is the junction where each branched end of the axon transmits information into another cell. Is that true? No. Why is that not true? Awesome. And glia cells are supporting cells that insulate the axon. Is that true? Awesome. So both A and C are going to be correct. And does anybody know the other function of our glia cells that we talked about in class? Awesome. So glia cells also nourish the cell. All right. So depolarization can be described as which one of those? Any ideas? All right, so is depolarization a reduction in the cell's membrane potential due to sodium ions exiting the cell? No, awesome. Is it an increase in the cell's membrane potential due to sodium ions exiting the cell? No, awesome. Is it a reduction in a cell's membrane potential due to sodium ions entering the cell? Are we getting more positive or less positive when we are in our depolarization? Less positive, more positive? We're in the inside of our cell, we're getting more positive. Because remember, our resting potential is negative, and then we have to get more positive for our action potential. So a reduction in the cell's membrane potential due to sodium ions entering the cell, is that going to be true for depolarization? That is true. So we're reducing the cell's membrane potential because more of our sodiums are entering the cell to make it more positive. Is everybody good with that? Okay. Cool. All right. So gated ion channels. Which one of those is true about gated ion channels? So do gated ion channels open and close due to changes in membrane potential? Yes, awesome. And specifically, what type of gated ion channel is that one? Is it going to be voltage gated or ligand gated? That's a description of our voltage gated ion channels. And do gated ion channels open due to binding of chemicals to the ion channel? Is that true? That is true. So that's going to be an example of our ligand-gated ion channels. And do gated ion channels open and close sporadically on a timed schedule? No, right? Awesome. So D is going to be our answer for that one. Any questions about that one? No? Okay, cool. All right. So which of the following regarding the sodium-potassium pump is correct? Which one of those is right in terms of the sodium-potassium pump? Does the sodium-potassium pump work so that for every 2 potassium ions that enter the cell, 3 sodium ions leave the cell? Is that one correct? Awesome. So we know that that one cannot be correct because that's the opposite. And is the sodium-potassium pump energy dependent? Awesome. So our answer is going to be both A and C, and we're still in chapter 48, I think. Yeah. Any questions about that? Which of the following is not a stage in information processing? Okay, so is sensory input a stage in information processing? Awesome. So our sensory neurons transmit information from our sensors detecting external stimuli. And what about interneuron transfer? Is that a stage in information processing? No. So this one is not. Is integration a stage in information processing? Yes, awesome. And what about motor output? Is that a stage? Awesome. So this is our only one that's not a stage in information processing, and that's still chapter 48. All right. If two excitatory synaptic inputs were received with a long break in between the two, what would you expect to happen? So would you expect membrane potential to rise with each input, but not enough to reach the threshold, so action potential would not be generated at the axon hillock? Does that sound right? Maybe. So what is the hillock? What is the axon hillock? So our axon hillock is part of the neuron, obviously, and it's going to be our integration center. And if action potential is going to reach the axon hillock, what does that depend on? So whether or not action potential reaches the axon hillock depends on whether it's excitatory, inhibitory, and the temporal and spatial summations of all of the different inputs. So let's go through these. So the summation of two excitatory inputs would cause membrane potential to rise to the threshold, and action potential would be generated at the axon hillock. So what do we think about that one? Let's look back at the question. So we're saying that there's a long break in between the two excitatory synaptic inputs. So if there's a long break between the two, they're not going to be able to sum together to reach the threshold for the axon hillock. So we would not think that action potential would be generated at the axon hillock. So membrane potential would lower with each input, but not enough to reach the threshold, so action potential would not be generated at the axon hillock. And we know that one's incorrect. Why? Because they're excitatory, right? So if they're excitatory, we're not lowering the membrane potential, we're raising the membrane potential. And we can say the same thing for this one, because that also says the membrane potential is going to lower. And the membrane potential would neither rise nor lower, so action potential would not be generated at the axon hillock. The excitatory inputs are going to have an effect. It's just whether or not they have enough of an effect to reach the threshold to generate an action potential. So that one's going to be false. So the only one that's true of those is that the membrane potential would rise with each input, but it would not rise enough to reach the threshold. So action potential would not be generated at the axon hillock. Does that make sense to everybody? Okay. All right. So which of the following is false? All right, so nurse equilibrium is the point at which the concentration gradient of an ion is equal to the opposite electrostatic gradient. Is that true or false? Awesome. That's going to be true. Ions cross the membrane down a concentration gradient by passive diffusion. Is that true or false? True. Awesome. Ions diffuse from the side of the membrane at which they are least concentrated to the side of the membrane in which they are most concentrated. Is that true or false? Awesome. And so ions cross the membrane across an electrostatic gradient. That one's going to be true. And the four most important ions to maintain Nernst equilibria are potassium, sodium, calcium, and chlorine. And that one's going to be true. So this is going to be our only false statement, and that's still in chapter 48, I think. Yeah, perfect. All right, is everybody good with that one? Okay. What else can we say about the four ions, these four ions? Mm-hmm. Awesome. So they all have different charges, and they need to balance the ions across the membrane, and they each have their own permeability. They're not equally permeable to the membrane. Yeah. Okay. All right. What structure in the mammalian brain functions selectively, controls which products enter and leave the brain in order to maintain a stable chemical environment? White matter, gray matter, meninges, ventricles, or the blood-brain barrier? Blood-brain barrier, awesome. So what can we say, what do we know about our white matter? Awesome. So it's mainly made up of axons. And where is it located? Awesome. Inside of brain, outside of spinal cord. And what do we know about gray matter? What's it made up of? Awesome. And what do we know about the meninges? Awesome. Connective tissues that protect the central nervous system. And what do we know about ventricles? Awesome. So they're fluid-filled spaces. And they're continuous with the central canal of the spinal cord. Awesome. And that's chapter 49. All right. So number 26. The autonomic nervous system can be divided up into what categories? So is the autonomic nervous system divided up into central, sympathetic, and parasympathetic? No, that one's not going to be our correct answer. Is it divided into the peripheral, sympathetic, and parasympathetic? No. What about peripheral, parasympathetic, sympathetic, and enteric? Yes. Does everybody agree with that? That's a yes? What's enteric? Can anybody answer that? What does the enteric control? Awesome. So our enteric controls muscles and glands of the digestive system. So C is not going to be our correct answer in this one. What about central, parasympathetic, sympathetic, and enteric? That one's not going to be our correct answer either. So our autonomic nervous system is made up of the parasympathetic, sympathetic, and the enteric. And can anybody tell us, is the ANS, is that part of the peripheral nervous system or the central nervous system? Awesome. That's going to be part of our PNS. And what does the parasympathetic, what is that in charge of? Awesome. So in the short, our short little reminders, I guess, of those are that the parasympathetic is rest and digest, and the sympathetic is fight or flight. So this is energy conservation, hence rest and digest, and the sympathetic is energy consuming activities. Is everybody good with that? All right. So what is the function of the medulla oblongata? Does our medulla oblongata assist with our breathing? Awesome. Does it assist with our digestion? No. It does assist with our digestion. And does it assist with our balance? No. Awesome. So it assists with both our breathing and our digestion. Does anybody know the other two things that it also helps with? So it also helps with circulation and swallowing. And what about balance? What part of the brain helps with balance? Awesome. This is going to be our cerebellum. And there's also one more piece that helps with breathing. What's another part of the brain that helps with breathing? Also, awesome. So our breathing is both the medulla oblongata and pons, and the cerebellum is with balance. And where are the medulla oblongata and the pons located? And they're going to be in the hindbrain. Awesome. Any questions about that? Okay. So which area of the brain is responsible for voluntary movement and cognitive functions? Is it our cerebral cortex? Any ideas? So our cerebral cortex is going to be responsible for voluntary movement and cognitive function. That's in chapter 49. All right, and awesome. Any questions about that? Okay, so which of the following is false? Multicellular organisms evolved from simple recognition and response processes. Is that going to be true or false? That's an easy one, guys. Did we evolve from simple recognition and response processes? Yeah. Animals with bilateral symmetry tend to have more specialized nervous systems. Is that going to be true or false? That's true, right? The human brain development shows approximately the same developmental path as the evolutionary transition from fish to tetrapods to mammals. Is that true or false? Awesome. So what does that pathway look like? So the hindbrain shrinks and the forebrain grows. Does that make sense? So when we're developing, then that's what happens as we develop, and that's the same thing that happened in evolutionary history. Does that make sense? Okay, cool. Slow-moving and sessile animals have relatively simple sense organs but show a high level of cephalization. Is that true or false? False, awesome. So that's going to be our correct answer. And that is false because they have little to no cephalization. Awesome. Any questions about that? Okay. What symptoms would you expect to be associated with damage to our parietal lobe? So, would you expect to see reading disabilities if we had damage to our parietal lobe? Awesome. Would you expect a loss of hearing? No. Awesome. Would you expect a loss of balance? No. And would you expect blurred vision? No. Awesome. So A is going to be our correct answer. So other than reading, what else is our parietal lobe? What else is our parietal lobe responsible for? So it's reading, speech, and taste. And what region of the brain would you expect loss of hearing? Where would you expect damage from that to be? Awesome. So the temporal and the frontal lobe both have hearing functions. And what about loss of balance? Awesome. So the hindbrain or the cerebellum. Yeah. And what about blurred vision? Awesome. So, any questions about that? Okay. All right. So, what methods have researchers used to discover information on the brain? So have researchers used symptoms and deficiencies in patients with brain injuries? Yes, awesome. Have they used PET scans? And what about MRIs? Awesome. We use all of the above. And within the cerebrum, there is a band of nerve fibers that connects the left and right hemispheres. What is that structure called? I heard some words, but I did not hear anything clear. Awesome, C. So that's going to be our corpus callosum. And what is the disorder of when the corpus callosum is severed? What disorder do we get? Awesome. And most of these are made up, but the basal nuclei, what do we know about the basal nuclei? That's a real thing. Awesome. So you're correct. So it's in the cerebrum, and it's important for motor coordination. And do we remember what disorder what disorder comes with damage to the basal nuclei? Awesome, Parkinson's. Okay. Any questions about that? Okay, cool. Which of these modalities corresponds to hair cells used for sensory reception? So, is that going to be our visual? Do our visual use hair cells? No. What do our visual use? What type of what type of receptors? I just said it. So, photoreceptors. Is it going to be tactile? What is tactile linked to? Awesome. And so that would be mechanoreceptors, right? And do we use hair cells for auditory, for hearing? Awesome. And what about chemical? What receptor are we talking about if we have chemical? Awesome. And what about electric? Awesome, electroreceptors. And this is where chapter 51 questions start. These were all chapter 49. Any questions about that? No? Okay. So a caterpillar making a cocoon is an example of what type of behavior? Awesome. It's going to be innate behavior. So how would we describe innate behaviors? Like, how would we define them? Mm-hmm. Awesome. So there's some sort of environmental trigger or cue or part of development where it just happens. You just automatically do it without having to learn it. Awesome. Any questions about that? Okay. Which of the following is an example of operant conditioning? So the Pavlovian response of dogs trained to drool when they hear a bell due to its association with food. Is that operant conditioning? No. What type of conditioning is that? Awesome. Rats who were given food when they pressed a lever learned to press the lever immediately when they grew hungry. Is that operant? Awesome. And duckling bonding with their mother and following her around during their youth. Is that operant? No. What is that? Awesome. Awesome. So it's like a trial and error. And operant conditioning is usually associated with reward and punishment. So any questions about that? That's a good question. So why isn't it considered classical conditioning? What do we think? So with the operant conditioning, it's trial and error with associated with reward and punishment. And with classical conditioning, you're just associating two unrelated things with each other. So this is classical conditioning because they rang a bell at the same time as they gave the dog food, and the dog already drools when it sees food. So it's not a learned behavior for the dog to drool. It's just that they're conditioned to drool when they hear the bell ring because it's associated with food. But with this, it's a learned behavior. They learned to press the lever because that reward was associated with it, not just because somebody pressed a lever while they were giving them food. Does that make sense? Okay. Any other questions about that?
So operant is learned behavior and classical isn't?
So that's not totally correct. No, it's the difference between it's the difference between trial and error and just association, basically. Does that make sense? Yeah. So classical is more of association. And this is, they learned that when they pressed the lever, they got food. They're not given food at the same time as the lever is being pushed, so they know to be hungry at that point in time. Does that kind of make sense? Okay, cool. Any other questions about that? Okay. So I think there's an E here. So in sexually dimorphic communities, one sex often displays conspicuous traits or behaviors that can make them easier targets for predation. Why would these traits persist through evolutionary time if they seemingly lower the fitness of the individuals? All right. So what do we think about that one? All right. So do we think that mutations have not arisen to get rid of the harmful traits? Does that make sense? No, that does not make sense. Mutations have arisen to get rid of the harmful trait, but they are linked to heritable diseases that are even more harmful to an individual's fitness. What do you think about that one? No, perfect. When intersexual selection is at play, the choosy sex often prefers individuals with apparent handicaps. Is that true? Awesome. Sexually dimorphic organisms are usually at the top of the food chain and do not need to worry about predation. Is that true? No. And these traits used to be more extreme and more conspicuous. Natural selection is a slow process, but will eventually eliminate these traits. Does anyone think that one is true? No, awesome. So why is it that in intersexual selection they often prefer individuals with apparent handicaps? Because if that individual is able to survive with that handicap, it means that they're a strong competitor, right? They're a strong individual. They must have good genes if they're able to survive if they have that handicap. Does that make sense? Okay, cool. And that's still chapter 51, I think. Yes, that's still chapter 51. Right. sequence of unlearned behaviors directly linked to a simple stimulus is a, what do we call that? Is it a fixed stimulus pattern? Awesome, B. So a fixed action pattern. And what is a sign stimulus? A sign stimulus triggers the fixed action pattern. And once you have that behavior, that fixed action pattern, are you likely to get rid of it, or is it likely to stay with you? Awesome. All right, any questions about that? Okay, digger wasps are known to use landmarks to locate their nest. If the landmark is moved, they will fly to the landmark rather than the nest location. What is this an example of? Associative learning, simple association, simple learning, spatial association, or spatial learning? E, awesome. That's still chapter 51. All right. So, in what conditions is parental care often limited? Is parental care often limited when young require intensive parental care? Or, sorry, I read that wrong. That says paternal, not parental. So in what conditions is paternal care often limited? When young require intensive parental care, it is more beneficial for males to conserve energy by mating with many females than to help one female care for his offspring. Is that true? B and C, awesome. So, when young do not require intensive parental care, it is more beneficial for males to conserve energy by mating with many females than to help one female care for his offspring. And when it is difficult to determine paternity in cases like external fertilization, are both cases when you would think paternal care would be limited. Awesome. There was also, I don't know if he fixed it or not, but there was a typo in the study guide that says it's more difficult to determine paternity with internal fertilization, but that should be with external fertilization. So he has it correct in the lecture notes, but not in the study guide for chapter 51. He may have changed it, but I'm not sure. Any questions about that? No? Okay. A scuba diver finds a shell on the ocean floor in a region where no sunlight is penetrating. What can be said of the location of the shell? So would the shell be in the paleagic and aphotic zone? No. So the ocean floor, what zone is that going to be? C, D. So our ocean floor, that means we're going to be benthic. And our paleagic is like the open water, right? So we're on the ocean floor, not in the open water, so it can't be paleagic. And where no sunlight is penetrating, is that going to be aphotic or photic? Awesome. So it's going to be in the benthic and aphotic zone. Any questions about that one? No? Okay. In what region would you most likely find coral reefs? So what do we know about coral reefs? Are they going to be an intertidal benthic zone with nutrient poor water? Where is our intertidal zone? So I'm going to draw a really horrible picture. Forgive my horrible artistry. So if we have, like, that's our beach, then our intertidal zone is going to be up here, right? So we can't have coral reefs where we're in our intertidal zone. And is it going to be in the neritic benthic zone with nutrient poor water? Or neritic benthic zone with nutrient rich water? So it's going to be C, because we know that they're going to be in tropical from our biome chapter, so chapter 52. We know that coral reefs are found in tropical, shallow, and nutrient poor water. And our neritic zone is going to be right here. And this would be our paleagic. So it's going to be right here with nutrient poor water. Is everybody good with that? All right, so blank are groups of different species living in the same area at the same time. These occur in blank, which include the biotic and abiotic elements in an area. Is that going to be populations and ecosystems, populations and communities, communities and ecosystems, ecosystems environments, or communities environments? Awesome. So communities are different species living in the same area, and they occur in ecosystems that include the abiotic and biotic elements of an area. All right, so which of those is an example of a biome? Awesome. So why are those other ones not biomes? What makes C a biome? Awesome. So our biomes are going to be globally similar ecosystems. All the broadleaf evergreen trees worldwide, trees aren't an ecosystem. Abiotic features aren't an ecosystem. Oceanic vertebrates are not an ecosystem. So coral reef ecosystems are a biome. Awesome. Any questions about that? Okay. You are in an area with cold winters, hot summers, and a lot of rainfall. You see many deciduous trees. What type of terrestrial biome are you in? Tropical rainforest, tropical dry forest, temperate broadleaf forest, northern coniferous forest, or temperate grassland? Any ideas? Awesome. So we're describing our temperate broadleaf forest biome. How would you describe your tropical rainforest biome? What was that? Yep. Awesome. So it's on the equator. What else can we say about our tropical rainforest? What about the temperatures? Awesome. Are you going to have a lot of rain? Awesome. And what about our tropical dry forest? How is it going to be different from our tropical rainforest to have a tropical dry forest? Awesome. It's in the name, right? And so what about our northern coniferous forest? What are those going to look like? So we're in the north, so what do we think about our winters? How are our winters going to look? So we're going to have long, cold winters and short, wet summers. And what about temperate grassland? What is our temperate grassland biome going to look like? I'm going to have fire. What else are we going to have in our temperate grasslands? What are our temperatures going to be like? So think about what your temperate broadleaf forest is like. So your temperate broadleaf is going to be cold winters, hot summers, and a lot of rainfall with mainly deciduous trees. So how would our temperate grasslands be different? Awesome. So the main thing about our temperate grasslands, different from our other temperate biomes, is that they're going to have frequent disturbances and fires to knock out the trees. So it's all going to be that pioneer communities. You're in an area with mild, wet winters and dry, hot summers. You see mainly shrubs. What type of terrestrial biome are you in? Awesome, chaparral. And so we all know what desert biomes are like, right? It can be hot, not a lot of rain. Awesome. Awesome. So we're going to be hot, low rain. And how would a savanna be different from a desert or a chaparral? So this is going to have seasonal rain and grassland vegetation. We already said that this one is going to be drought and fires. And what about a tundra? Awesome. Cold. All right. Is everybody good with those ones? Which of the following correctly describes an aspect of global patterns? So photoperiods vary greatly across latitudes worldwide. Is that true? Do our photoperiods vary greatly? No, except for in the poles. The photoperiod, what is the photoperiod, first of all, actually? So our photoperiod is looking at the entire year, how many hours of daylight you average per day. So over the course of a year, at most latitudes, everywhere gets about 12 hours a day if you average it. So obviously seasonally that's going to change, but your photoperiod is looking at the entire average. Due to its high heat capacity, water heats up faster than land. Is that true? No, right? So land heats up more quickly. Mountains influence precipitation, not participation. Precipitation patterns by blocking wind, which allows precipitation to accumulate all around the mountain range. Is that true? So our mountains do influence precipitation patterns, but you're going to see rain shadows. So you're not going to see precipitation accumulate all around the mountain range. You're going to see that everything gets blocked here, so you're going to be dry on this side. Does that make sense? Do we remember that part? Okay. The heat in the tropics leads to rising bands of moisture which release precipitation. The resultant dry air lowers to form bands of aridity on either side of the equator. Is that true? Awesome. So that's our one true statement. So D is going to be our correct answer there. Is everybody good with those? Chapter 52. And these have all been 52 with the biome stuff. Is everybody good so far? Okay. You have discovered a new species of rodent, and you determine their burrows are uniformly distributed throughout your research area. Which of the following is a reasonable conclusion you could draw about this population of rodents? So would you say that their distribution is independent of resource availability? So think about your different patterns. You have uniform, random, and clumped, right? So what do we know about uniform distribution patterns? You probably would not think that their distribution is independent of resource availability, right? So if it was independent of resource availability, you would more likely see a random or a random pattern. Interspecies interactions affect their local distribution. So could we say that? So interspecies would be like the rodents with another type of species that's not their species. So can we say anything about interspecies? No, right? We don't really have enough information to say if that's a factor. Intraspecies interactions affect their local distribution. So if they're uniformly distributed, would we expect intraspecies interactions to affect their distribution? We would, right? And if new populations of this species are found elsewhere, their burrows will have the same spacing pattern. What do we think about that one? So we can't generalize. We can't generalize that just because we found a population that's uniformly distributed in one area, that if we find the same species in another area, they're going to be facing the same environmental factors. So we can't make the assumption that all of that same type of rodent are going to be uniformly distributed. So that is going to be C, and this is where chapter 53 starts. All my questions. Okay, is everybody good with that one? Okay, cool. What type of survivorship curve would you expect to see for sea urchins which have a high mortality rate early in life, but if they survive this period, they are unlikely to die? So do you remember our survivorship curves? We had our 3 survivorship curves. Which type of survivorship curve is this one? So this is going to be your type 1, your type 2, or your type 3. This is going to be our type 1. So what type of organisms have a type 1 survivorship curve where they have a high survivorship early in life, but then once they reach a certain age, they're probably going to die? So that's going to be like us, right? So type 1s would be like humans and organisms with a lot of parental care most of the time. Be cautious though if I don't think he would ask you a question like this, but within species there can be exceptions. Like in human populations, there are different populations that see a type 3 survivorship curve, even though we generally say that humans have a type 1. So if he gives you a species and then describes it, just make sure that you're not going just based off of what you normally see with that type of species. Make sure you read the entire question, because there are exceptions to the generalizations that you use. And so this would be our type 2, where it's pretty equal all throughout. And this is our type 3, where you have a high mortality rate early in life, but if they survive until a certain period, then they're not likely to die for a while. Is everybody good with that? Okay. And that's also chapter 53. All right. So interpret the life table. So is it true to say that an individual has an 86.5% chance of surviving to age 2? Yes? Does everybody agree with that? So we have to look at the difference between these two columns. So if you're looking at this column, what are you computing? What's the difference between those? If you're looking at this column, you're saying this is the likelihood that these individuals will survive through just that 1 year. So there's an 87.6% chance that 0-year-olds are going to live to be 1-year-old. So there's a 86.7% chance that 1-year-olds are going to live to be 2-year-olds, and there's an 86.5% chance that 2-year-olds are going to live to be 3-year-olds. So this one is saying of surviving to age 2 in general, and that's what we would look at this second column for. So this one is computing the chance that you're going to live up to age 1, age 2, age 3, age 4. So an individual actually has a 76% chance of surviving up to age 2. Does that make sense? An individual has a 29.7% chance of surviving to age 4. Is that one true? Awesome, that one's true. And an individual has a 65.7% chance of surviving from age 3 to 4. No, what's the percent chance that an individual has to survive from age 3 to 4? Awesome. So they have a 45.2% chance of surviving from age 3 to 4. Okay, is everybody good with that? You get the difference between those? Okay. So What is not a correct statement about the cohort represented by the life table in problem 49? So, females wait until they are 3 years old to breed, will have a higher overall reproductive fitness than those who start breeding at 2 years old. Is that true? So that is not true. Why is that not true? So we're saying with this average number of offspring per female, we're not saying anything about her future clutch sizes. So just because a female breeds at age 2 and has 5, that doesn't mean that it's necessarily going to reduce the size of her clutch if she breeds again at age 3. So this table is only saying that the average number of offspring per female for that age group is 5, 8, 6, and 2. It's not saying anything about how it would lessen a female's fitness in future years if she breeds. So that one is not going to be true. It may be, I mean, when you get to ecology, there are different life tables that show like how breeding 1 year might affect your future clutches, but with these ones, it's not saying anything about that. The life table is depicting one population at one point in time. Is that true? No. Why is that not true? So what is a cohort? So our cohort is following the same group of individuals from year to year. So they fake tracked these, this same 283 for 6 years until they all died. So this is all the same from the same birth year. Like if this was 2010, this is 2011 for the same age group. We're not adding anybody from any different age groups. So it's not one point in time, it's following them throughout their lifespan. And the maximum age of this cohort is 5 years old. Is that true? Awesome. So our answer is that D, both A and B are incorrect. Is everybody good with that? Okay. Which of the following is false regarding logistical growth? What do we think? Does carrying capacity decrease as the population size increases? No, right? Because our carrying capacity is a set number. It doesn't matter what the population size is. It's always going to have that carrying capacity unless some sort of environmental factor affects the resources available. When the population size is low, growth rate increases with population size. Is that true? That is true, because if we remember our little graph, if that's our carrying capacity, we're going to have something that looks like an exponential growth until about halfway there. So when our population is low, then our growth rate is going to increase with population size, but when it hits that tipping point, then it's going to decrease with population size. Okay. As the population size nears carrying capacity, the resources available per individual decreases. Is that true or false? That's true, right? Because if we have more individuals, then there's not as many resources to go around. And that's kind of the point of carrying capacity, is that there's only a certain number of resources available. And does anybody remember your logistical growth equation? No? dN over dt equals Rn times your k minus n over k. So it's the exponential growth equation with this little carrying capacity problem multiplied onto it. Okay. Are there any questions about that? No? Okay. What factor is not considered when calculating the change in population size over 1 year? Immigration, emigration, carrying capacity, birth rate, or all of these factors are considered? Awesome. So what is our equation for calculating the change in population size over 1 year? So NT plus 1 equals the population size at T plus the birth rate minus the death rate plus immigration minus emigration. So carrying capacity only comes into play when you're looking at your logistical growth model and you're actually looking at your growth. Is everybody good with that? Okay. So which of these interactions is an example of, oh, whoops, skipped one. Which of the following is not an abiotic factor that affects species distribution? Precipitation, soil composition, temperature, or competitors, or all of those affect? What was that? Awesome, competitors. Because those are a biotic factor, right? So which of these is an example of symbiosis? Predation, mutualism, parasitism, both B and C, or all of the above? Awesome. So both B and C are correct. And what is the third type of symbiosis? Awesome. And this is where we started, chapter 54 questions. All right. So the idea that two species competing for the same limited resource cannot coexist is called what? What do we call that? Is it the competitive exclusion principle, the competitive restriction principle, character displacement principle, local restriction principle, or species limitation principle? Awesome, thank you. So the idea that two species competing for the same limited resource cannot coexist is the competitive exclusion principle. And that's also chapter 54. And what is a possible result of niche overlap? Expansion of a fundamental niche, expansion of a realized niche, resource partitioning. I'm losing my eloquence here. My mouth is too dry. I can't talk right anymore. So which one of those is a possible result of niche overlap? So what is a fundamental niche? So the fundamental niche is the hypothetical area that an organism could exist, and that decreases when you look at the realized niche because of different factors like competition. So if you have niche overlap, that's not going to affect the fundamental niche, because in niche overlap what we're saying is that two organisms, like the bivalve example that he had, is that two organisms are in the same area and their niches are overlapping, so they're going to be competing in the same area where they're overlapping. So we can't have a fundamental niche expansion, and the realized niche is not going to expand because there's going to be one competitor that's either going to outcompete the other organism, and their realized niche would just stay the same, and then the other one would reproduce or reduce its realized niche. So we're not going to have any expansion of a realized niche. One of them could have a reduction of a realized niche, or both of them could have a reduction of a realized niche. But what we would see potentially is resource partitioning. So with resource partitioning, we're saying that they're finding a way to use the resources available differently, even though they have the overlap of the niche. Does that make sense for everybody? Okay, cool. And two species have similar beak shapes when they occur in different areas, but when they occur together, the average beak size of one species decreases while the other species' average beak size increases. This is an example of which of those? Is it an example of competitive exclusion? So it is going to be an example of competitive exclusion. Is it also character displacement? Awesome. And is it convergent evolution? No. What is convergent evolution? Do we remember? So convergent evolution is when two species evolve similar traits independently. So without any shared ancestors, because of shared environmental conditions, they separately evolve the same trait. So this is not an example of convergent evolution, because we're just seeing a way that they're using character displacement to divide the resources. So character displacement, resource partitioning, and local extinction are all possible outcomes of competitive exclusion. Is everybody good with that? Okay. A non-toxic species of frog is colored similarly to another brightly colored toxic salamander in the same environment. This is an example of which of those ones? Awesome. Batesian mimicry. And is everybody good with that? Okay. Red mangroves protect the shoreline soil from erosion, and many organisms utilize their underwater root system for protection. Red mangroves are an example of dominant, energetic, keystone, primary, or secondary species. Awesome. So they're going to be an example of a keystone species. Why wouldn't they be an example of a dominant species? So dominant species are going to be abundant, like the most abundant in the area. And dominant species can also be keystone species, but we're not saying that the red mangroves are abundant in any way. We're just saying that that many organisms are utilizing their underwater root system. So the only thing that we can say about them is that they're a keystone species. And the intermediate disturbance hypothesis states that moderate levels of disturbance lead to the highest species diversity. Why is this the case? What do we think? Okay, so if there is too little disturbance, dominant species will outcompete other species. If there is too much disturbance, slower growing species will never get established. Is that true? Awesome. So A is going to be our answer for that one. And that is chapter 54. Okay. Are there any questions about that? All right. If I could get you all to do this survey evaluation. It's just about the study union. It's not about any past SI sessions that you've been to. Are there any questions? Mm-hmm. 19. Yeah, 19, the correct answer is C. Yeah, so if you think about like you're depolarizing, so you're going from -70, so that's your higher membrane potential, and then you're adding the positive ions, so you're increasing in your positive charge. So the potential of that membrane is reducing. So like if you think about it then, if you think about it in the way that your resting potential is normally like your highest membrane potential amount. So it's kind of the opposite. Like if you think about your signs, you're kind of going opposite than would normally be considered like reducing and increasing. Does that make sense? Yeah. Okay. Are there any other questions?